Hello boys, welcome back to another interesting session of video tutorials, and this session is meant for class 11th biology students, and this is meant for 18th August 2020. Our memory helps us in reflecting upon what we have been analyzing, studying and reflecting upon. As you remember, we were discussing about glycolysis. For clarity's sake, I am using different form today instead of the regular PPT, and we need to calculate how many molecules of ATPs. ATP, I said, energy currency, adenosine triphosphate molecule. So we need to arrive at the possible 38 or 36 ATP molecules that are produced on oxidation of one glucose molecule. So for that sake I have taken this, and also it serves as a revision for you. So glycolysis, glyco means glucose, lysis means breaking. One molecule of glucose is being broken. So what is the use for us when one molecule of glucose is broken? And we are thankful to three German scientists, Emden, Meyerhoff and Parnas. That's why the glycolysis has got another name as EMP pathway. E stands for Emden, M stands for Meyerhoff, P stands for Parnas. So it's also called EMP pathway. So this occurs, as I said yesterday in our previous session, cytoplasm of the cell, and is a universal pathway in biological system. In this, one molecule of glucose is reduced to two molecules of pyruvate. So this is the main step that you must remember in glycolysis. One molecule of glucose is reduced to two molecules of pyruvate. And the first step in this is glucose is phosphorylated to glucose 6-phosphate by ATP in the presence of an enzyme called hexokinase, alpha glucokinase and Fg2+. Second step, glucose 6-phosphate is changed to its isomer fructose 6-phosphate with the help of enzyme phosphohexose isomerase. Step number 3, fructose 6-phosphate is further phosphorylated by means of ATP in presence of an enzyme phosphofructokinase and Mg2+. The product is fructose 1,6-diphosphate or biphosphate. The irreversible phosphorylation reaction catalyzed by phosphofructokinase is the rate limiting step in glycolysis. This controls the entire process of glycolysis. It is controlled by the concentration of substrates, ATP and fructose 6-phosphate. Next, fructose 1,6-biphosphate or diphosphate splits up enzymatically to form one molecule each of 3-carbon compound glyceraldehyde 3-phosphate. So we say 3-phosphoglyceraldehyde, PGAL, say 3-PGAL. And another molecule is dihydroxyacetone 3-phosphate, DHAP, and the enzyme involved is aldolase. So fructose 1,6-biphosphate is split into two molecules, 3 carbon each. One is PGAL, 3-PGAL, 3 stands for 3 molecules of carbon. Another one is DHAP. Now DHAP is isomerized to glyceraldehyde 3-phosphate by an enzyme triose phosphate isomerase. Now next, in the presence of enzyme glyceraldehyde hydrophosphate dehydrogenase, glyceraldehyde 3-phosphate loses hydrogen to NAD to form energy-rich compound NADH2 and accepts inorganic phosphate to form 1,3-diphosphoglycerate. Next, one of the two phosphates of diphosphoglycerate is linked by high energy bond that can synthesize ATP and form 3-phosphoglycerate in the presence of an enzyme phosphoglycerate kinase. So the direct synthesis of ATP from metabolites is called substrate level phosphorylation. Now 3-phosphoglycerate is changed to its isomer 2-phosphoglycerate by an enzyme phosphoglyceromatase, which transfers the phosphate from third to second carbon. Though through the agency of the enzyme enolase, 2-phosphoglycerate is converted to the final product of glycolysis, phosphoenol pyruvate, PEP, phosphoenol pyruvate. Now the high energy phosphate of 2-phosphoenol pyruvate or pyruvic acid is transferred to ADP by the enzyme pyruvate kinase to generate 2 molecules of ATP per molecule of glucose oxidized. The molecular form resulting from the loss of the high energy phosphate is pyruvate. So 1 molecule of glucose is broken down to form 2 molecules of pyruvate. Now in glycolysis, since ATPs, 2 ATPs are used and 4 ATPs are produced. So gain of ATP is 4 ATP minus 2 ATP, so net gain is 2 ATP. 2 molecules of NADH are formed at the time of oxidation of glyceraldehyde 3-phosphate to 1,3-diphosphoglycerate, and each NADH is equivalent to 3 ATP. Therefore, the net gain of ATP in glycolysis is 2 into 2 NADH into 3 ATP. So 2 3 is 6 ATP plus 2 ATP produced. 6 plus 2, 8. So the net gain of ATP in glycolysis is 8 ATP. So you must remember this. This is an important step. So because of glycolysis, we are getting, every cell rather gets 8 ATP molecules. The net reaction of glycolysis we can write this way: glucose plus 2 NAD plus 2 ADP plus 2 H3PO4 because phosphorylation. 2 pyruvates because 1 glucose is forming 2 pyruvate or pyruvic acid molecules, and NAD, 2 NAD becoming 2 NADH plus H plus. 2 ADP is becoming 2 ATP. So this 2 NADH, each NADH is equivalent to 3 ATPs. So 2 NADH, 2 into 3, 6 ATP. 6 ATP plus 2 ATP, 8 ATP. That's how we are arriving at the conclusion that glycolysis results in the formation of 2 molecules of pyruvates and 8 molecules of ATP. Now, next step, after glycolysis, there is an intermediate step, that is oxidation of the pyruvate. Pyruvate, the end product of glycolysis, enters mitochondrial matrix. You see, glycolysis occurs in cytoplasm of the cell. Now it is entering into mitochondria. Through a specific transport protein, it is decarboxylated oxidatively to produce CO2 and NADH. The product combines with sulfur containing coenzyme A to form acetyl-CoA. So acetyl-CoA is a coenzyme. The reaction occurs in the presence of an enzyme complex, pyruvate dehydrogenase, and of course Mg2+. This step is called gateway step or link reaction because acetyl-CoA acts as connecting link between glycolysis and Krebs cycle. So what happens in this link reaction or gateway reaction? Pyruvate, 2 molecules of pyruvate, remember, and NAD plus CoA in the presence of pyruvate dehydrogenase and Mg2+, what's happening? Acetyl-CoA is formed. This is very, very important. Without acetyl-CoA, 2-carbon chain, coenzyme, there is no Krebs cycle. For Krebs cycle to take place, acetyl-CoA is a must. And that NAD is becoming NADH. So pyruvic acid to acetyl-CoA, 2 molecules of NAD, because 1 pyruvate molecule, 1 NADH, 2 pyruvate molecules, 1 more NADH, so 2 NADH. Each NADH above I said worth 3 ATP. So 2 into 3, 6 ATP molecules are produced in this link reaction. Okay, you must remember. So glycolysis 8, right? And link reaction, pyruvate oxidation, 2 NADH, 2 into 3. So 6 ATP. So 6 plus 8. How much? 14 ATPs are produced as on now. Now Krebs cycle, phase 3 in this. So citric acid cycle. The Krebs cycle is also known as citric acid cycle. This was thanks to Hans Adolf Krebs in 1937 based on the studies of oxygen consumption in pigeon breast muscle. Krebs cycle operates in the mitochondrial matrix. You must remember this for your UPSC NDA. Mitochondrial matrix, the powerhouse of the cell. That's why mitochondria are called powerhouse of the cell, because it contains complete mixture of various respiratory enzymes. Now this acetyl-CoA 2-carbon compound combines with oxaloacetate, a 4-carbon compound, in the presence of condensing enzyme citrate synthase to form tricarboxylic 6-carbon compound. That's why it is called tricarbo TCA cycle. It is called citric acid or citrate. It is the first product of Krebs cycle. CoA is liberated. Citrate or citric acid undergoes reorganization in the presence of aconitase, forming cis-aconitate and water. Now this is the dehydration. Now this is how it happens. As you are seeing, pyruvate 1 NAD, and since 2 pyruvates 2 NAD, we have already calculated. This is a link reaction, right? And this acetyl-CoA is becoming citrate. Citrate has become cis-aconitate. And cis-aconitate is converted into isocitrate with the addition of water in the presence of an enzyme aconitase. This is hydration. You can see H2O we have put inside. And now isocitrate is dehydrogenated to oxalosuccinate. Now oxalosuccinate is formed in the presence of an enzyme called isocitrate dehydrogenase and Mn2+. And this is very important step for us because energy rich compound NADH2 is produced. Next, oxalosuccinate, which is unstable compound, is decarboxylated to form alpha ketoglutarate, as you are seeing here, alpha ketoglutarate through the enzyme decarboxylase. This step releases 1 molecule of carbon dioxide, as you are seeing. So 6-carbon oxalosuccinate has become 5-carbon alpha ketoglutarate. Now alpha ketoglutarate is both dehydrogenated with the help of NAD+ and decarboxylated by an enzyme complex alpha ketoglutarate dehydrogenase. So you can see here this step also is very, very helpful for us because it is again giving us NADH2, energy rich compound, and we are very much interested in this because 1 molecule of NADH2 is equivalent to 3 molecules of ATP. So very highly energy producing step. Alpha ketoglutarate is becoming succinyl-CoA. So remember, alpha ketoglutarate is 5 carbon and succinyl-CoA is a 4 carbon. So decarbox, 1 CO2 is out. And succinyl-CoA, the product combines with CoA to form succinyl-CoA. To continue the cycle, succinyl-CoA is converted to succinic acid by the enzyme succinyl thiokinase or succinyl-CoA synthetase. This reaction requires GDP, and this in the presence of inorganic phosphate. The high energy bond of succinyl-CoA is transferred to GDP, guanine, this ATP we said adenosine triphosphate, like that here GDP. Now this succinate again, succinyl-CoA to formation of succinate also is energy forming reaction. And now succinate 4 is becoming fumarate. Succinate is also called succinic acid. Succinic acid is metabolized to fumaric acid or fumarate by dehydrogenation. 1 molecule of water is out, fumarate. And again, this is very, very energy rich compound, FADH. 1 molecule of FADH is equal to 2 molecules of ATP. Again, this is energy forming reaction. So succinic acid is metabolized to fumaric acid by dehydrogenation, catalyzed by succinic dehydrogenase, as you are seeing. It is the only dehydrogenation reaction in the citric acid cycle that involves the direct transfer of hydrogen from the substrate to FAD without the participation of NAD. This is the first step where FAD is getting hydrogenation. So FAD is converting into FADH2. So there it is, FADH2. A molecule of water gets added. See, in other reactions you have noticed water is removed in some place, but here from fumaric acid to malic acid, water is added in the presence of fumarate. So malate is formed, and when fumarase becomes malate, malate is dehydrogenated or oxidized through the agency of malate dehydrogenase to produce oxaloacetate. So we are back to the circle, oxaloacetate. And oxaloacetate picks up another molecule of activated acetate to repeat the cycle, as two molecules of acetyl-CoA are formed from one molecule of glucose. So total 24 ATPs are produced from one acetyl-CoA. Net equation of Krebs cycle is 2 acetyl-CoA, because it is one link reaction. If you recollect from your memory, link reaction has made pyruvic acid to form acetyl-CoA. Since 2 pyruvic acids, 2 acetyl-CoAs. So that's how 2 acetyl-CoA plus 6H2O. You can see in all the reactions where added, water is added, and 6 NAD plus 2 FAD plus, okay, FAD plus, and 2 GDP and inorganic phosphate. So what happens? 4 CO2, 2 H2O and 6 NADH, and each NADH is equivalent to 3 ATPs, right? So 6 into 3, 18, and FADH2, 2 molecules, 2 into 2, 4, 18 plus 4, 22, 22 plus 2 GTP, 24, 1 GTP is equal to 1 ATP. That's how we are arriving at net calculation of 24 ATP molecules, okay? Now, this is the overall net reaction of TCA cycle or Krebs cycle. So total in 3 steps. In glycolysis step number 1, we got 8, right? And in link reaction, we have got 6. 8 plus 6, 14. And in Krebs cycle, 24. So 24 plus 14, 38. So that's how one molecule of glucose will give us on oxidation 38 ATP molecules. And next we go to the fourth phase, electron transport chain, in our next class. And just write this Krebs cycle once, and you will have to remember first products, like how you will remember pyruvate acetyl-CoA, both together you write citric acid, citric acid to cis-aconitic acid, cis-aconitate to isocitric acid, oxalosuccinic acid, alpha glutarate, then succinyl-CoA, succinic acid, fumaric acid, malic acid, and oxaloacetate. So first you try to make a ring or a circle with the products. Then what you do, write the enzymes at each step. Then you come to whether water is added, whether carbon is added, carbon is removed, NAD is formed, FAD is formed. At least I want you to remember where energy is formed, like isocitrate to oxalosuccinate, NAD becoming NADH2. Alpha ketoglutarate to succinyl-CoA, NAD is becoming NADH2. Succinyl-CoA to succinate, ATP, or you can write GDP plus inorganic phosphate becoming